engineering C1 metabolism in bacteria. Nico graduated his PhD in 2017 and then moved to work at the Max Planck Institute for Molecular Plant Physiology as a 4-year postdoc, where he accomplished the unique feat of managing to engineer methylotrophy in a non-model bacteria. He is now an assistant professor at Wageningen University, where he works on continuing engineering C1 metabolism. Nico, please take us away.
Yes, thanks a lot, Will, for the nice introduction. Also nice to see all of you, or at least see some of your names and faces. Sorry, I didn't make it to London this time, but I'm very excited to give the talk anyway, and hopefully next time can also be there in person. So today I will talk about rewiring metabolism towards what I call a sustainable C1 lifestyle, and I will later make clear why in bacteria. So in my postdoc and also in my current group, we're doing a lot of synbio and metabolic engineering in different type of bacteria. And I will today show some of this work to give you a flavor. So first, with some small acknowledgement, or actually big acknowledgement also to Aaron Baravan, who was instrumental in many of this work because he was my postdoc supervisor. Unfortunately, he passed away briefly after I left his lab, but yeah, I owe a lot of thanks also to him for all the work I can present today and that we're still building on, and also his group that is still connected well. Will also used to be part of that. So unfortunately missing in the picture on the right because you were ill, I think, at that reunion, which was last summer. But just an acknowledgement to start with. Good. Then let's go into the science. And first, I always like to start a bit more general. Why are we doing these things that we're doing? And so the vision behind that is that in the end, we like to produce our carbon-containing chemicals from the most abundant ultimate feedstocks, so the ultimate renewable feedstocks that we can think of, which are water, carbon dioxide, and renewable energy. And these feedstocks are in principle fully scalable, unlike any other waste or biomass resource you can think of, which are of course also interesting sources for sustainable bioproduction, but these are really ultimate sources. So chemistry, chemical catalysis, and especially electrochemical catalysis, has been demonstrated in the last few years to be very effective in reducing CO2 with electricity into small mediator molecules like carbon monoxide, formic acid or formate, methanol, as well as the production of hydrogen from water, which of course does not take CO2. So chemistry is very good in doing this left part, making the small molecules. And the efficiencies, the energy efficiencies of these processes, as I will show you later, are extremely high. The rates are pretty high. These are like cathodes, electrochemical systems where you can do this at high rates, which is way more efficient than the energy conversion, for example, by photosynthesis into chemicals. So we really believe that chemistry really has an edge there to make these small mediator compounds, but then to go from these mediators into bioproducts, biomass, chemicals, all kinds of molecules you can think of. Biology seems to be the best solution. And so what we envision is that you could combine chemistry and biology in so-called hybrid systems where you generate first these mediators from electricity and CO2 and water if needed, and then use these mediators to feed the bioprocess. So if we look into this concept a bit further, and we looked at this from the perspective of energy efficiency, first and foremost, then we first looked at chemical catalysis. So we teamed up for this work a couple of years ago during my postdoc with also an electrochemist, Dennis Cupriavidus, as well as Charlie Cotton, fellow postdoc, and Aaron Baravan and myself. And we did an extensive analysis back then of what is the current status of chemical catalysis and also biological catalysis, of course. And so if you look at chemical catalysis towards these simple molecules like hydrogen, you can see on the left, and then carbon monoxide, formic acid, as well as methane, methanol, and ammonia. All of these can be made at energy efficiencies about 50%. So if you put in electrical energy, you get back about 50% of that energy in the form of chemicals. And that is really high if you compare what biological photosynthesis typically does is that you find back less than 1% of the energy that's put in the form of light. So if you would couple this 50% to a solar panel, which typically has an efficiency of around 20-25%, so if you take 20% of 50%, you still have 10% of your energy into these molecules, while photosynthesis normally ends up with 1%. So you can make chemical reduced compounds at a much higher efficiency from renewable energy than what photosynthesis can do. So that's our starting point. You see that hydrogen can be made best, or at least at the highest efficiency. This is also technology that's scaled up already. The electrolysis of water is already being done for the production of green hydrogen in some places, and will definitely also fuel probably the future so-called hydrogen economy that also industry is probably going to rely on more. And maybe also some vehicles might be fueled by hydrogen made in this way with an energy efficiency of 70 to 80%. But also the one carbon carriers like carbon monoxide, formic acid can be made at reasonably high efficiency, around 50%, and methane, methanol, and ammonia can be made in a two-step process at very high efficiencies as well. And especially for methanol, this is also already being considered for scale up. For example, in Iceland, they're building huge plants where they make methanol from renewable electricity. Actually, this goes via in between step with hydrogen. Anyway, that's just the current status in terms of energy efficiency, or it was, let's say, 3 years ago when we published this. By now, some of these efficiencies have even gone up a bit more. But that's all great and good, but then we go to the next step, and then we go to biology, and then it gets a lot more complex, as biology always does. So biology gives us a lot of options to convert these molecules, the ones I showed you previously that can be made by chemistry, into biomass and products. If we look at the upper efficiency range, the dark green bars, they're all anaerobic organisms. So here we're talking about so-called acetogens. So anaerobic organisms that use the Wood-Ljungdahl pathway, or also known as the reductive acetyl-CoA pathway, which is strictly oxygen sensitive, but is very energy efficient, that can convert, for example, methanol, formic acid, hydrogen, or carbon monoxide into acetate, or also ethanol and some other molecules. And this can be done at efficiencies up to 90%, especially also for formic acid and methanol. And this is very interesting, or already being commercialized for the use of hydrogen and carbon monoxide coming from steel mills. So the LanzaTech process, that's probably some of you know, which is, I would say, one of the most impressive biotech companies out there that managed to leverage these organisms to make bioethanol, but also improving portfolio of products in recent years. They have demonstrated at very high efficiency. This is very promising, but these acetogens also have major drawbacks. As they can only grow anaerobically, they are also very limited in what they can make. Not all products can be made. Products that need high investments of ATP in their biosynthesis cannot be made efficiently by acetogens. So you cannot make full biomass or proteins, let's say, at a high efficiency. You could maybe make a bit of it and co-produce a lot of ethanol or acetate, but if you really want to make that as a product, you need to go aerobic. And if we go to the aerobic organisms, so this is, we looked into natural organisms. We scanned the whole literature going back from the 60s of last century till today, and we looked into all reports of yields, basically how much biomass do you get from consuming a certain amount of substrate like formic acid or methanol. Then you can calculate their energy efficiency. And that's what we did for a whole list of data points. And then we came to these bars, which cover the spread of the values. And then you can see that dependent on the assimilation pathway that these organisms use, they have different efficiencies. So on the higher end, around 50% energy conversion efficiency from formic acid, for example, to biomass. In this case, we looked at biomass yields for these aerobic organisms. You see efficiencies of around 50% for the serine cycle. The serine cycle is one of those few C1 assimilation pathways known in nature. For methanol, we see similarly high efficiencies for what is called the RUMP, or the ribulose monophosphate pathway. Those are definitely very promising and relatively efficient. But if we look on the right side of the spectrum, we see, for example, the Calvin cycle organisms. So we see again formic acid and the hydrogen on the Calvin cycle. And the Calvin cycle is also notorious for being relatively inefficient because it takes quite a lot of ATP. And these, in the end, these organisms, also if we measure their yields, what is reported in literature is a lot lower. Around 30% of the energy in the mediator is conserved into the final biomass. And then if you look into ammonia, for example, that's also via the Calvin cycle, that it fixes CO2. It has a very low efficiency, so we even do not further consider that as a realistic option, even though ammonia can be made via chemical catalysis relatively efficiently. So if we look to the C1s, there is potential, and also to hydrogen, but aerobically, there is challenges with the assimilation pathways, especially if we don't use the rump or the serine cycle. So that was one of the takeouts from our extensive literature analysis. If you combine these numbers together from biological, chemical and biological catalysis, you can have the complete energy conversion potential of electromicrobial production, as we call it. And you see, obviously, that the aerobes do worse than the anaerobes, and hydrogen typically comes out best because it has a really high chemical conversion efficiency. And on the aerobic side, where I took the best pathways for hydrogen, methanol, and formic acid, there is room for improvement. And that's what we're working on, the aerobic side of things. So in a nutshell, the key challenges in aerobic bioconversion are that we need more efficient bioconversion of C1 substrates to product in terms of energy, in terms of stoichiometries. And another challenge that I haven't mentioned yet, but is the tolerance to substrates and products. So growing on, for example, formic acid or methanol is not super trivial for every organism because they are substrates that are related to toxicity. Hydrogen is another interesting option, but that brings explosion risks and gas transfer issues in your reactor. So it's very interesting, but also has some downsides there. So the soluble substrate methanol and formate are very interesting in my perspective, but they're also toxic, especially formic acid. Actually, it's more toxic for microbes than methanol typically. So that's some of the challenges. So when we're looking into biocatalysis using whole organisms as catalyst, we basically are looking for super microbes and super pathways, super C1 assimilation pathways in this case. And so if we look at organisms, we can think of characteristics like how well it can make products. Are there maybe already organisms out there that are great for a certain product? Or other organisms that already have a high tolerance to formic acid, or maybe tolerance to a product we want to make. And I think in general, as I like to do with synthetic biology, it's important, genetically accessible, but also to combine these properties and to improve properties of substrate consumption and product formation. It's good to have genetically accessible organisms with decent or developing toolboxes. Then if we look on the pathway side, so we really try to be open to any pathway design before we started into these kind of projects. We say we're not bound even to a naturally existing pathway like the Calvin cycle or the serine cycle. We can dream of any pathway, and then we try to dream of fast pathways with good kinetics, so overall pathway kinetics, meaning that the enzymes in the pathway on average are fast enough, that the ATP consumption is low, and that there is no wasteful electron reactions. I will show you an example of that later, and a reaction where really electrons get dumped, which is a very wasteful process. So we try to find pathways that have this low ATP consumption, are fast, and don't have wasteful steps in terms of electrons. Of course, there's trade-offs. The perfect pathway doesn't exist because often ATP is used to drive reactions to become faster. So there's trade-off, for example, between energy consumption and speed to some extent. But there seems to be interesting pathways out there that we might not even have found in nature yet. So one of these pathways that was actually designed by Aron Bar-Even already over 10 years or about 10 years ago is the so-called reductive glycine pathway. And when he described this pathway for the first time, it was described as a synthetic pathway. This pathway is was back then not known in nature. And basically the pathway consists of all natural enzymes. So the enzymes are out there in nature in other pathways and in various kind of organisms, but the architecture of this pathway specifically was not found yet. So the pathway starts with formic acid. So it was primarily designed as a formic acid assimilation pathway. So this interesting one carbon substrate that is soluble. And this simple acid, this one carbon acid, is assimilated into methylene THF via activation with 1 ATP and a cofactor tetrahydrofolate, and then it's reduced to methylene THF. This is what we call the C1 module or C1 activation module. And this module is actually also found back in the Wood-Jungdahl pathway, so the very efficient pathway which operates in anaerobic acetogens. So they share this pathway module. But after that, the architecture of the pathway is slightly different, or quite different. So from that stage on, once you generate methylene THF, the methylene THF is further reduced into glycine, and that's why it's called the reductive glycine pathway. And so in this step, CO2 is fixed. So it combines CO2 with methylene THF to make the C2 amino acid glycine. And this is done by the so-called glycine cleavage system that is basically operating in all of us, in all living organisms we know, but it predominantly cleaves glycine in most organisms, generating methylene THF, for example, for DNA biosynthesis. But this complex is fully reversible. Thermodynamically, it's very close to equilibrium, so it's fully reversible, and you can push it if you have enough CO2, so you need high CO2 and methylene THF, you can push it to make glycine. And that's what is done in this pathway. So then this glycine is combined with another methylene THF. So this first module has to run twice. And this second methylene THF is condensed with glycine to generate serine, a C3 amino acid. And C3 serine can be deaminated to pyruvate. And then we're at the heart of metabolism, from where we can go to any biomass precursor or product precursor we wish to go to. So this is a relatively simple pathway, very linear in its nature, unlike most natural C1 assimilation pathways, which are very cyclic. And it's only about 10 steps. And in these 10 steps, you only spend 2 ATP, only in the first reaction, which you have to run twice because you have to make 2 methylene THF. Apart from this assimilation pathway, you also need to invest more energy to reduce formate up to the level of pyruvate, because formate is very oxidized substrate. So some of the formate you actually need to oxidize through a formate dehydrogenase to generate NADH, and this NADH can be invested within the pathway, and some of it can also be used to generate this small amount of ATP you need. So basically you need to run these 2 modules in parallel. So this is the general pathway design. I hope, by the way, things are clear. If not, please interrupt me. I can see if you raise your hand via the Teams function. Then I will try to give your answer already while we go. And if there are more general questions, you can also keep them till the end. Good. Then we go next to a superhost. So at the beginning of my postdoc, we selected an organism in which we wanted to implement this non-existing pathway. And the organism we selected is pretty well known. It's called Cupriavidus necator, or also Ralstonia eutropha, which was its name that had been used for decades in literature. But as it goes with microbes, sometimes scientists decide we need to change their names. So the current name is Cupriavidus necator. And Ralstonia is known for several reasons. One of it is that it can make polyhydroxybutyrate, a bioplastic naturally at very high titers. Basically, up to 80% of its biomass, as you can also see on the picture, can be these granules full of bioplastic. And they're made when the organism experiences nutrient starvation, like phosphorus or nitrogen starvation. So the organism actually already has been used in industry in the past to make the bioplastic polyhydroxybutyrate. Also, the genetic toolbox was developed. It is a natural formator trough, but it uses the Kelvin cycle, and it also has a reasonable formate tolerance. I say reasonable because it's not that tolerant. It's about 100 millimolar, which may sound like a decent amount, but realize it's only a C1 molecule with low energy, so 100 millimolar is not that much. It's comparable, let's say, to 20 millimolars of glucose, roughly, or actually even a bit less in terms of carbon and energy. If you add more, it really starts to suffer from it. So you really have to do a fat batch if you want to go to higher densities. But it can grow in it, and it has a decent tolerance. E. coli, for example, can naturally only tolerate about 30 millimolar. So this organism already has some history of production. It has already formate tolerance. It has a good formate dehydrogenase already, which is a challenging enzyme to engineer because it's a complex enzyme, a metal-dependent multi-subunit enzyme, but it uses not an efficient pathway with the Calvin cycle. By the way, ralstonate can also grow on hydrogen. I won't talk about that further today, but it can also grow on hydrogen via the Calvin cycle. That's also what it's very well known for. So it's already a well-known, let's say, C1 and CO2 fixing organism. So it has this Calvin cycle. So basically what it naturally does, it oxidizes all formate when it grows on formic acid to CO2, all of it. And this CO2 is reassimilated into biomass via the Calvin cycle, via a CO2 fixation pathway. And in the process of doing that, it takes quite a bit of ATP. It takes about 7 ATP to make one pyruvate. One pyruvic acid takes at least 7 ATP from the Calvin cycle. And that is pretty energy intensive. If we look to this reductive glycine pathway, which you can see on the left that I already introduced to you, this is just a different drawing of the same pathway, it takes 2 ATP to make one pyruvate, so a lot less ATP. And if we then could calculate the theoretical yield of pyruvate per 10 moles of formate, we can see that we make about 35% more pyruvate per 10 formates or per formate in general than the Calvin cycle could. That's a theoretical prediction. Then we also see that if we would model biomass formation and we ignore maintenance, which is of course not a very correct assumption, but that was the easiest to calculate, we expect the yield could also be about 20% higher, which will probably be a bit less because of this maintenance, which ATP that needs to be spent either way if you grow on the Calvin cycle or the reductive glycine pathway. But there is a clear gain to lift the production of biomass and potential also products from formic acid by replacing this Calvin cycle by the reductive glycine pathway. But of course, replacing such a cycle, which is really the central metabolic pathway if you grow on formic acid, could be quite an endeavor. But we were up for it, and that's what I started working on in my postdoc a couple of years ago. So the way we followed, and this was a typical, or is a typical way of engineering this kind of pathways in the lab of Arend Bar-Even, is to couple modules, so sections of the pathway directly to growth. And this is a really smart strategy because it allows you to select for working modules and also evolve them if needed without needing to do a lot of complicated measurements because we can just select the fastest growing organisms. And how do we do this? So for example, for the first modules, or basically the first 2 modules, where we convert formic acid into glycine, we made a glycine oxytroph Cupriavidus necator strain. So we knocked out its native pathways for glycine biosynthesis, which can both be from serine and from threonine cleavage. And so there were 3 knockouts needed in this strain, and this triple knockout strain cannot grow unless you feed it with glycine into the medium. And so this glycine oxytroph is a perfect platform to select for the activity of the first couple of non-natural enzymes. I say non-natural here, especially for the first part. This is true because the conversion of formate to methylene THF, the first enzyme is Cupriavidus necator lacks completely, and for the second one, it doesn't have an efficient candidate. So what we did is we took those enzymes from another organism, from Methylobacterium extorquens, which uses those naturally for the serine cycle, in fact, and we expressed them on a plasmid library. And at the same time, we overexpressed the native glycine cleavage system, which it natively uses to make some of its C1 from glycine. But now we want to have a major flux to it, so we overexpressed it on a plasmid. And we made libraries for both these modules with different constitutive promoters. So we had developed a library of constitutive promoters for Cupriavidus necator to allow different strengths of expression of these enzyme modules, to see where is the sweet spot of sufficient expression, sufficiently active expression without overburdening the system. We did this initially on plasmids, as you can see, and we made a whole library of plasmids, and we could easily test those plasmids into this glycine oxytroph strain, and we identified a combination of 2 plasmids with actually a very weak promoter for the C1 module and a bit stronger promoter for the C2 module, which allowed good growth without glycine, but on formate. That's the green line in the graph. So basically here we convert formate into glycine because we can feed formate. We have both plasmids in and we get a decent growth rate, similar to the brown line, which if you would feed glycine. So we can really reconstitute this auxotrophy by having a functional pathway from formate to glycine. We also perform labeling to really confirm that this pathway is happening. So we fed C13 labeled formic acid. That's what we typically do. And then we look at the proteinogenic amino acids. So the amino acids in proteins, we lyse the proteins. And then we look at glycine and serine, for example, as you can see in the bar plot on the right. And you see that glycine is labeled almost fully once because one of the carbons of glycine in this pathway is coming from formate, and the other one is coming from CO2 and the glycine cleavage system, or glycine synthesis actually in this case, it's incorporating CO2, which is unlabeled in this experiment. Serine is non-labeled because there is a knockout between glycine and serine in this oxytroph, and histidine is also labeled almost fully once, and histidine gets one carbon from formyl THF, one of the intermediates in the pathway. Also coming from formate. So there are a confirmation that the pathway is working. So glycine and C1 precursors in this strain are already coming from formate, so we have the first part of the pathway established. Then we go on to the next challenge, is to get the next module working, basically. And we went bold here. Basically, we said maybe Cupriavidus necator has already the ability to convert glycine into biomass, which is the rest of the pathway, basically. And we found, and actually I'm not showing these details here today, but you can find it back in the paper that we published. Cupriavidus necator has a native route to assimilate glycine, but we only found it after evolution, short-term evolution of the strain. Actually, natively it didn't grow on glycine, but after a few weeks it did. And this route went via glyoxylate, not the route that we had in mind to engineer this glycine assimilation to proceed via was via serine to pyruvate. Here, glycine instead is oxidized to glyoxylate via glycine oxidase, and basically here electrons are dumped. And that's an inefficiency in its native glycine metabolism that we discovered. And this is an example of what I mentioned in the beginning of a wasteful electron step. And electrons are very expensive. If you really donate the electrons to oxygen, which happens in this step, you're basically throwing away a lot of energy, a lot more than wasting 1 or 2 or 3 ATPs, but actually 2 whole electrons. So basically 2 whole formate molecules. One formate molecule. Anyway, this is what it natively does. So when we grew these strains with the previously selected plasmids, the P1 and the PC2, and we introduced this into a Kelvin cycle knockout strain. So this is not the glycine oxytroph anymore that I talked about in the previous slide, but this is a Kelvin cycle knockout. So it cannot grow in formate at all. It's knocked out for rubisco, the core enzyme of the Kelvin cycle. Both of its copies are knocked out, and this strain cannot grow unless we add the 2 plasmids and we also add formate, and that's the purple line you can see. But this growth only happened after short-term evolution and was very slow. It has a doubling time, as you can see here, of almost 60 hours. So the student that did this experiment literally left his tubes in the incubator, forgot about them, and came to me a few weeks after and said, Nico, we've got growth on formic acid. He didn't expect this actually anymore because it was very slow. You can see this is when we repeated this experiment in a plate reader to measure the growth. You see that the experiment lasted over 200 hours. So really many days experiment. So very slow. And also the yield is very low. If you look to the black line, that's the wild type strain on the Kelvin cycle. So it has a higher yield, it is much faster. So it's far from what we promise. It's not a better pathway, it's actually worse, it's very slow. And one of the reasons is that it takes this detour where it wastes electrons. So what we then did is we wanted to reroute this, but first we moved, because we wanted to use more plasmids for this initially, but we didn't have too many compatible plasmids and we didn't want to overburden the cell with 3 plasmids. So we moved the expression of this glycine cleavage system, which was first on a plasmid, on the genome by introducing a strong constitutive promoter in front of its native glycine cleavage system. And that is this what we call CAG2 strain, which grew actually slightly better, showing that this genome-based expression gave a bit faster growth, about 45 hours doubling time COG2, but still a low yield. And then we made the big jump, and that's what you can see here in COG3. The next strains, so these are all COG3 strains, there we really see a much higher yield and a faster growth. And what we had to do for that was only one thing. We basically overexpressed the root via serine. Actually, both enzymes are natively present, but C. necator did not overexpress these enzymes from glycine to serine and from serine to pyruvate. We overexpress them both on a plasmid, and we see very much better growth on formic acid and CO2. Still, the competitive route was there, but we knocked out some of the steps in this route, and we saw actually that it hardly made a difference in growth. Actually, it grew even a bit better while it was still there, but it was not really significantly different. So we showed that we rerouted this route, and that, let's say, the inferior natural route for glycine via glyoxylate was not really an issue anymore once we introduced the more efficient route via serine. So this was a nice rerouting that really improved the growth phenotype a lot. But still, we were far from what the Calvin cycle can do. Our yield is lower, and also our growth rate is lower. We left it for some short-term evolution, and we got COG4, the 4th generation Cupriavidus reductive glycine pathway strain. But even that COG4 was still not that fast compared to other strains. So then that was basically where my postdoc finished. We published this rerouting of metabolism to the new pathway, which completely could take over the Calvin cycle, but not beat it yet. And then Bo Dronsella in Arendt's lab took over the work. He started his PhD, and Bo took up the challenge of introducing the whole pathway into the genome, which is something that I didn't do yet, because we started with plasmid-based engineering of most of the modules. Only this middle module, the glycine cleavage system, was already expressed from the genome with a constitutive promoter, but the rest was still other two modules, C1 and the C3, the bottom one from glycine via serine to pyruvate, was still on plasmids. So what Bo did is, because genome engineering tools in Cupriavidus necator are not that great, we don't have a CRISPR system working yet. We have been trying hard, but we don't have it working yet for counter selection. So it's good old recombination, and it's a bit annoying, the system. So what Bo said, why not use transposons? Because transposon was described to work well in Cupriavidus necator. Their random integration into the genome is actually a useful tool in this case, because we can just identify good spots for integration, because we can select for the best locations. So what Bo did is he used the transposon to integrate first the C1 module into the genome in any place, and then did a competitive growth experiment and he did this for 10 sequential cultivations on formic acid to fish out those cells that integrated C1 module into the genome and led to fast high yield growth. And then he repeated the same trick, so then he made COG5, as we called it, and then he repeated the trick for the 3rd module to also get that into the genome, and then he ended up with strain COG6, which has all the modules in the genome. And to our nice surprise, I would say, this is what we hope, but it's always hard to have too high expectations in biology. This COG6 strain for the first time could beat the yield of the Calvin cycle, as we already predicted based on its higher ATP efficiency. And we see about, I think it's a 13% higher yield of biomass on the same amount of formic acid for the strain. So this was really nice to us, because this was the first time that we can show that a synthetic pathway that on paper should perform better than the natural one in an organism can really beat it. However, unfortunately, it cannot beat it yet in terms of growth rate, as you can see on the graph on the left. The purple line of COG6 grows around 11 hours doubling time, while the wild type doubles every 6 hours. So it's a bit slower, but high yield so far, but this might be improved further. We have a preprint on this work, and there you can also find more proteomics characterization of the strain and some more details on bioarchive. So here I end my Cupriavidus part of the story, and then I will give you a bit broader view on our work and the work of many others in this field of engineering synthetic C1 assimilation, as some of you are maybe also active in, like I know Will is working on this as well. So to give you an overview of the reductive glycine pathway, the pathway I've talked about so far. We demonstrated it in Cupriavidus necator. We now show it can even beat the yield of the Kelvin cycle. Other work of Arend Barreven's lab 2 years ago also demonstrated this pathway fully in E. coli. E. coli can naturally not grow in formic acid, but by introducing this route, they made it grow in formic acid. And they also have shown, Arend's lab had also shown part of the pathway to work in Saccharomyces cerevisiae already some years ago, but only the first part of the pathway. The full pathway is not yet established in Saccharomyces cerevisiae, which is, of course, would be very interesting if we can get yeast to grow efficiently on formic acid. And also Pseudomonas putida was very recently demonstrated to have partial growth on the pathway, both in work by Pablo Nical's group, that's the paper of Justine Tolin, which was actually accepted in metabolic engineering a couple of weeks ago. So it still says bioarchive here, but it's in metabolic engineering now. And we also in parallel did an effort in Wageningen to make Pseudomonas putida use the reductive glycan pathway, also to grow on CO2 and methanol. I will show you that in a couple of slides. So the pathway is expanding to different types of organisms, but none of these works, even the upper ones, which are most advanced in terms of getting the full pathway to work, getting full formative trophic growth, have shown real growth rates and yields that are up to industrial performance, I would say. So there's still a way to go, and I believe this could be both achieved by a combination of more genome-wide rational engineering, which we're currently doing in my lab, and also laboratory evolution to further get faster growing high yield strains. So principles are there, several organisms, but still work to be done. Then as a small intermezzo, we called this a synthetic pathway or a new to nature pathway, the reductive glycan pathway, but actually more or less around the time that we started working on it, a colleague of mine in Wageningen, Irene Sanchez, she discovered that this pathway also may operate in a natural organism, and we proved this 2 years ago as well via several omics techniques. And actually this is an anaerobic organism, sulfate oxidizer, so it generates energy via the oxidation of sulfate, or the reduction of sulfate rather than oxygen. And this organism can grow on hydrogen and CO2, so real CO2 fixation, and it had none of the known CO2 fixation pathways that were so far known in literature, but it had all the genes for the reductive glycan pathway, which is maybe not so surprising. Many organisms have those genes, but in this organism, we could really demonstrate also via metabolomics with labeled substrates and proteomics and transcriptomics that it really uses this reductive glycan pathway, and we could officially prove its existence into nature. However, it was not exactly the same variant via serine that I've been talking about before, but rather a variant that reduces the glycine to acetyl phosphate and then to acetyl-CoA, which is an even more energy efficient pathway. It takes only 1 ATP because it takes another CO2 here and doesn't have to convert 2 formates into methanol and THF. But this variant is strictly oxygen sensitive, this pathway. And this pathway makes it actually energetically very close to the Wood-Ljungdahl pathway. So this was a very cool finding, also from a fundamental and an ecological and physiological perspective, that we could elucidate this so-called synthetic pathway to also exist in nature after we actually already engineered a similar pathway in the lab. Good. To convince you that the reductive glycan pathway could become a prominent pathway also for other substrates. So far I talked about formic acid mostly. I have this slide. So the reductive glycan pathway starts here normally with formic acid, but you can actually also add a step on top of the pathway where you reduce CO2 to formic acid. That's actually what this organism that I just showed you, this sulfate reducer with a natural reductive glycan pathway for CO2 fixation also does. They have reduction of CO2 to formate. They get the energy, for example, from hydrogen, and they use this redox power to reduce CO2 to formate. This is a challenging reaction thermodynamically, but it's possible. It's demonstrated in nature, it's possible. Another option to grow on is methanol, which is a very interesting C1 substrate as well. Methanol has a higher energy density than formic acid, has lower toxicity than formic acid, so and can also be made efficiently via electrochemical catalysis from renewable electricity. So it's a very interesting substrate. And methanol can be oxidized to formic acid, and in the process generate first formaldehyde and then formate. So this module, you could say the M module, you could also add on top. And this is what Kim et al. Showed in their 2020 paper already in E. coli, proof of principle for growth of methanol via the reductive glycan pathway. Very slow, 60 hours doubling time, but they could demonstrate that if you add this module to the pathway, it can work. The growth of formate in that paper was way faster, was around 12 hours, 10 hours, like we show Cupriavidus necator, but methanol, they could show only slow growth so far. But the proof principle is there. This is a versatile C1 fixation pathway for at least 3 different substrates, methanol, CO2, and formate. So it could be an interesting pathway, very ATP efficient for all of these substrates. So Leon Bruinsma, a talented PhD candidate in Wageningen University that I have co-supervised on this work, demonstrated this into Pseudomonas putida. So Pseudomonas putida, many of you might know, is I would say an increasingly popular model organism for bioproduction with a lot of tools and a lot of advances, and also can tolerate quite a lot of products. Justine Tolan also has shown that it has a high tolerance to formates, higher than E. coli, so that's also interesting. But what Leon specifically set out to is to add the methanol module, and he could show that again in an oxytroph, in this case a serum oxytroph, he could make it grow on methanol. So co-feeding glucose, but the serum is made from methanol. And he could also show via labeling that serum and histidine obtained their C1 carbon from labeled C13 methanol. So this was a proof of principle that we can make Pseudomonas putida into a methyllotroph, methanol consumer organism via the reductive glycan pathway, or at least the first module of it. This is not a full pathway yet. And even more excitingly, Leon showed for the first time that you can fix CO2 via the reductive glycan pathway in an engineered system. So in E. coli and Cupriavidus necator, we hadn't shown this yet, but in Pseudomonas putida, Leon took this serum oxytroph, added the first module, only the first module of the reductive glycan pathway, and showed if you add overexpressive formate dehydrogenase on top, so that's this thermodynamically challenging step, which Pseudomonas putida has a native metal-dependent enzyme that could do this at reasonable kinetics. He showed that he could grow on CO2. So part of the carbon here, as you can also see in the labeling, in serum and histidine, about half of the carbon is coming from CO2. And you can also see that if he added more CO2, so this is increasing from ambient up to 50% CO2, blue line, and you see that from about 20% CO2 there's good growth. So you really see that this phenotype benefits from high CO2 pressure, because this reaction, the first one, is really dependent on thermodynamics, so you need to push with a lot of CO2. But in an industrial biotech setup, this could be a reasonable concentration to work with. So this is the first step to engineer CO2 fixation via reductive glycan pathway. This only happened actually after evolution. I didn't tell you that, but in the first few weeks, again, no growth. Suddenly Leon saw growth, again showing the benefit of this growth coupled selection to identify beneficial mutations as well. And then Leon sequenced the genome, and he also re-engineered the mutation that he found, and one of the mutations was in NUOG, which is in the respiration complex of Pseudomonas putida, and we suspect that it inactivates or decreases the activity of this respiration complex. And we also show here the orange line that reconstituting this mutation at least partly explains the growth phenotype of the evolved strain. There's probably other mutations also having a smaller role, but this mutation alone can also restore the growth to a large extent. And this mutation probably helps to increase the NADH pool in the cell, because respiration is basically burning NADH into NAD. And this first step, the thermodynamically tricky step, you see the delta G at standard conditions is positive, so it means it would not be possible. So you can only make it possible by having both high CO2 and high NADH. And the CO2 example we already saw, it only works above 20% CO2, and works only well this growth, and probably also relies on this mutation, because the intercellular NADH pool will probably increase if you partly block respiration. So this was a nice demonstration of the combination, I would say, of rational and evolutionary engineering of CO2 fixation. So I want to end here. You see, it's almost 45 minutes. And so I hope I could convince you in this talk that the reductive glycan pathway is actually not a single pathway, but it's more like a family of pathways which can work for C1 assimilation of CO2, methanol, formic acid, and can assimilate glycine also via different routes. For example, the serine route, which seems the most efficient aerobic route to work with for now. But there's also other designs we can think of. And this pathway of families, I like to call the reductive glycan pathway variants. There's also other names out there now in literature because some other groups also worked on this pathway already in the past, but I suggest we use this name also as a small tribute to Arjen, who came up with the name reductive glycan pathway. So with that, I'd like to acknowledge all the people in my own group in Wageningen, all my former colleagues in the MPI in Potsdam, and a lot of collaborators across different universities in the Netherlands and internationally with whom I collaborate, and of course all the funding for making the research